So I'd like to give a few examples of how pathogens have evolved to evade humoral immunity. So there are some very successful pathogens out there that can infect many humans and keep infecting us, and we have a really hard time fighting and clearing these infections. So why is that? Why is the humoral immune system not perfect? A lot of different reasons. So here is a pathogen, and it's covered in some proteins, some sugars, and let's say we make a B cell with a B cell receptor with immunoglobulins that bind it. Well, that's great. We can activate, we can release antibodies, we're going to clear that infection, right? Sounds great. Not if that pathogen has many different versions. So there are different serotypes or different strains of pathogens. And that's why we can keep getting reinfected by the same type of pathogen over and over. An example is streptococcus, which pneumoniae, which causes lots of respiratory infections. So why do you keep getting infected by strep over and over? Why don't we have lifelong protection? Well, there are different strains or serotypes of the strep bacteria. So you might make antibodies against one, and a year later get infected with a different strain or serotype. And those antibodies are not good because the different strains or serotype have different shaped molecules on their surface. So you can keep being reinfected by a pathogen, not have any protection, if the pathogen has many different types of strains or serotypes. So the insides are more or less the same, the outsides are coated a little bit differently. There's also genetic variation that can occur on the surface of pathogens. So, for example, the flu virus, influenza, it's an RNA virus, and RNA has a high mutation rate in terms of it being a genetic material. So the proteins on the surface of RNA viruses, hemagglutinin and neuraminidase, those proteins can change. They can mutate. There can be antigenic drift in them. So this is why if you get a flu vaccine or you're infected by the flu and you survive, that's great. You make antibodies against that strain of flu. But a new strain of flu that might appear years later has different HA and neuraminidase molecules on their surface, so that the antibodies you made from a previous infection or from a previous vaccination will not help you fight this new infection. So anytime that a pathogen can change the molecules on its surface, it can allow that pathogen to reinfect the individual years later. So you have no protection against that. Another example of genetic variability involves a parasite called trypanosomes, and it's a parasite that causes sleeping sickness, found primarily in Africa. And this pathogen, right, we can make antibodies to it, that'd be great. There are molecules on its surface, glycoproteins, and sure, you can make antibodies to it. The problem is that the pathogen can change those glycoproteins. So these are actually called variable surface glycoproteins, and there are 1,000 different versions. So it uses actually gene rearrangement to shuffle its DNA to make different antigens on its surface. So that means that if infected by one of these parasites, you might be able to make antibodies to it, but then it'll just change its surface glycoproteins, and then you won't be able to make antibodies to it. So the fact that pathogens can change the antigens on their surface allows them to evade humoral immunity. Salmonella has 2 different versions of its flagella. So the flagella is the tail made of proteins. It can make 2 different versions. So if one is being attacked by antibodies, it just turns off that version, turns on the next version, and can evade the immune response that way. The pathogen that causes gonorrhea has these pili on its surface, but there are multiple genes that can form these pili. So if you make an antigen against one version of the pili, it shuts that gene off, turns on another gene, makes a different version of the pili. So anytime a pathogen can change what it looks like, it can evade the immune response. There are lots of pathogens which we have awesome ability to fight, either using our immune system or using vaccines, and it is due to the fact that the antigens on their surface are typically unchanging. So there are whole hosts of pathogens where we have excellent vaccines to, because these vaccines protect against all strains, and there's little to no mutation in the genes that create the antigens on their surface. So measles, mumps, smallpox, polio, rubella, these are typically unchanging in the antigens on their surface. So if you are protected against the measles, either you had the measles and you survived, or you were given the measles vaccine and had a good humoral immune response, you make antibodies to the measles virus, and those will protect you forever, because if measles comes back in 20 years, you've got protection from that, because your measles infection 20 years ago will still be recognized the measles that will come 20 years later. So, okay. What's next? One example of how pathogens can evade the immune system by immune evasion involves herpes viruses, and actually a number of viruses do this little trick to hide from antibodies. So if you're infected by a pathogen, such as herpes, obviously you're going to hopefully want to make antibodies that bind the herpes virus. And that would be good, because then you could neutralize the virus, you can opsonize the virus. But what herpes does to try to evade opsonization is it makes a virally produced FC protein. So you remember FC proteins, FC receptors that we make, those are proteins that bind to the FC region of antibodies. So for example, we have macrophages that have FC gamma receptors on their surface. Now, if this macrophage sees this pathogen covered in IgG, what's this macrophage going to do? It's going to go over there. The FC gamma receptor is going to bind the FC gamma region of those IgG, and that macrophage will phagocytose the pathogen. Awesome. But herpes makes this virus protein, and it covers up the FC region. So when this macrophage approaches this pathogen, there's nothing for it to bind to, because the FC region is covered up using a virus's protein. So the macrophage just swims by, doesn't eat anything. So this is one way by which viruses such as herpes can evade things like opsonization, by covering up the FC region using its own proteins, and that will prevent our own FC receptors from binding the FC region. So these are just a few examples of how pathogens can evade the immune system. There are many more. These are the ones I want you to know.